Hi, everyone, and thanks for joining us again for Phosphorus Science Now. I'm Matt Schultz with the Sustainable Phosphorus Alliance. Phosphorus plays a critical role in providing nutrition to plants and animals we depend upon for food, but much of it doesn't make it into our food. Rather, it remains in the soil and becomes what we call residual phosphorus. Figuring out how much residual phosphorus is stored in the soil, what forms it's stored in, and where exactly it resides turns out to be a very difficult, though very important task. Today's guest has written a review in the journal Global Change Biology about the challenges and importance of undertaking that task. We're honored to welcome back to the program Dr. Andrew Margenot of the University of Illinois at Urbana Champaign. Well, thanks, Andrew, for joining us again for Phosphorus Science Now. I'm very interested in hearing about this paper, so please go ahead.
Of course, thanks for having me back, Matt. So today I want to share with the audience out there on a recent paper that we published. This is a review paper. It's really more of a position piece where we integrate some data. And in this paper, led by the authors here in Global Change Biology, open access, by the way, we propose a way to think about residual phosphorus. And note that we use the word legacy phosphorus. In just a second, I'll talk about this disambiguation. But the whole crux of this paper is, how do we measure the phosphorus that's been accumulating in soils over the past 80 years since we began to increase the production of phosphate fertilizers? The co-author team is as follows. Shown here, I won't walk through the names. There's many of them. They're all great folks, and they all pitched into various aspects of the work. And thanks to the funders, Illinois NREC and US NSF for funding portions of this work. So a bit of background first to set the stage. We've all seen a similar graph like the one on the left, looking at, in those blue diamonds over time, the production of phosphate rock. This is the tonnage that's being mined. Also shown here, they like to depict world population, etc. But the idea here is that we have an exponential increase in phosphate rock mining and thus phosphate fertilizers post-World War II is what we often hear, but really it takes off by the 1960s. And in red here, we've got the projected demand by the Mosaic Company, a producer of MAP, DAP, and other phosphate inputs. So the idea is that we've exponentially increased the amount of phosphate rock mining, and this is consistent with estimates that we have quadrupled the amount of phosphorus flux to the biosphere. So biosphere here is being broadly defined as terrestrial and aquatic ecosystems, but the idea is that we've really revved up how much phosphorus is entering. It's an input flux. So this is a good piece that I'd recommend by Dr. Elser and Bennett, with one E, my apologies for that misspelling, on what this means for the biosphere and for the biogeochemistry of phosphorus. For reference, there's claims that we have doubled, maybe tripled, the flux of nitrogen through Haber-Boschan. So there's a relatively greater increase in flux for phosphorus, comparatively speaking. Now, we care about residual phosphorus because it's phosphorus that was not used, meaning it wasn't removed with crop biomass harvest or livestock for that matter. So think of this as a balance, input versus output. When the inputs of phosphorus, be it manure or fertilizer, exceed the export of phosphorus from the system defined as a plot, a field, a farm, a country, the world, or the soils of the world, better put. In any case, when inputs exceed the outputs, we have net accumulation. And this is a nice paper by Dr. McDonald, now McGill, that would depict here in the reddish colors a surplus, and deeper red would connote a larger surplus, but more specifically a positive balance, meaning more residual phosphorus in soils. Now, of course, phosphorus is the Goldilocks nutrient. We often have too little or too much. Think then of as the too much as this residual phosphorus built up in soils. Now, a word on the terminology here. I've been saying residual phosphorus, but a lot of folks like the term legacy phosphorus in soils. And in this opinion piece, it's just my opinion, I know this is contentious, we propose that we should probably stick to the origins of these terms. Legacy phosphorus was first being published in the 2000s in the context of Florida watersheds, referring to phosphorus that is fluxing in the watershed scale. Whereas the first use of residual was from Iowa in reference to previous fertilizer P applications. So that's why we opted to use residual phosphorus to refer to the soil scale idea or concept of this built up P from the past. So the overall motivating question of this paper was, how can we measure residual phosphorus? What are the ways that are available? Are there ways to combine these that would provide a net improvement in the estimate accuracy? And this is a concept figure from the paper, figure 1, where we try to map out the here be dragons of the world of phosphorus, and specifically of residual phosphorus. So you might imagine that as we go across scales of time and space, that there are some scales where it doesn't make sense or it's not possible to measure residual phosphorus. So imagine, the Pedon scale over 200 years. Do we have data sets for that? Probably not, or even archive samples. Same goes for very large scale spatially at short time frames. Is that meaningful? Maybe not. Most research, this is a qualitative proposition, tends to happen in this area. And then the blind spots we define as the area where we don't have much research, but it would likely be beneficial to invest more in quantifying the how much of residual phosphorus. The paper then synthesizes 4 major approaches to getting at how much residual P is there in a given system. Again, a system boundary can be from plot to field to country to globe. At first is mass balances. And in some sense, this should make sense to you because we oftentimes define, as I just did 3 slides back, residual phosphorus as a positive balance. So it stands to reason that we can use a mass balance inputs and outputs to define at a given scale of space and time the how much. The other approach is long-term experiments, field experiments where we are able to measure and monitor what's going in and what's going out, provide a very useful empirical basis for really adding with the first approach. We can construct high confidence mass balances that are less based on assumptions, often the case at large spatial scales, with measured input-output fluxes of phosphorus. Third is soil test P trends, basically agronomic tests based on extraction that we can use to estimate the directionality. So it's more of a qualitative. And what we found was that for a quantitative approach, they're not really that great for the how much. The fourth approach, borrowing from biogeochemistry, soil formation, is a chronosequence, where if you have a space for time substitution, you can estimate the accumulation of P in soils. So the proposition that we make in this review paper in GCB is the following. First of 3 is that any individual approach to measuring residual phosphorus is by nature constrained. There are simply pros and cons. Any single approach individually can't get the job done very well, and certainly not across spatiotemporal scales that are in high enough range. Second is that, well, here's a solution. They're often complementary. For example, mass balances pair very well with long-term studies. A chronosequence approach also pairs well with a soil test P trend if it's a field chronosequence approach, as an example. And the third proposition that we argue is that certain approaches are only feasible at certain spatiotemporal scales, meaning we have to come to grips with and make peace with the fact that there might be some blind spots that we simply cannot get at. This is really important because of the environmental and the policy implications. In other words, the uncertainty is front and center, a defining feature of how we should be thinking about residual phosphorus. I want to walk through just a couple examples of the approaches, including how we synthesize data to illustrate the pros and cons. So first, the mass balance is the gold standard, in part because we define residual phosphorus as a positive balance. Think of it as simply when the inputs exceed the outputs. There's a bigger number for the inputs. Now, this is, of course, a function of time. One can make a mass balance for phosphorus for, in theory, 1 day, but that's sort of silly or nonsensical. More likely is 1 season, 1 year rather, and then decadal, etc. Given the residuality of phosphorus, using that term in terms of its persistence, it would make sense to construct these at supradecadal timescales. Now, one thing to point out is that we find very few studies actually measure these small fluxes, either outputs like leaching, or the small input fluxes like atmospheric deposition. That may not matter, especially for systems that are very intensively managed with large input and large output fluxes. So the Corn Belt where I'm sitting, lots of P going in as fertilizer, lots of P coming off with grain. Even then though, in those high input output flux systems, these small, oftentimes unintentional, meaning that we don't manage them on purpose, these small fluxes can over time accumulate, and there can be amplification of error in the mass balance. Very few studies measure all of the inputs and all of the outputs. They oftentimes dismiss these smaller ones as, well, small in magnitude. This is an example of a mass balance that we just saw, I think as the fourth slide, showing the work by Graham McDonald, a mass balance at a global scale. And again, this is dependent on the time scale. And we can, of course, make these down to the field scale. And here's an example of that from the Morrow plots. The Morrow plots is housed at the U of I at Urbana Champaign, the second oldest study in the world, established in 1876. And we've got good data on yields and on fertilizers beginning in the early 1900s, when they first were being added, 1904. So what we can see here, we've got 3 of our 24 treatments. And beginning in 1876, the experiment started, but we've got yield data in 1888. And all 3 systems, which are shown here, have a negative balance in the early years. But it's a small negative balance because we've got very low yields because we're not fertilizing corn. This is all for corn yields. And then as we see the introduction of phosphate rock, 1910 through 1920, we see a very large positive balance. We see a large residual phosphorus as estimated by the balance. So again, we calculate a balance, we measure the residual P, preferably in soils. And a few points to make here is that the value of the balance, or rather the magnitude, can happen in very short time frames. And so the duration of the study doesn't necessarily help you predict the magnitude of how much for the residual P. The other is that uncertainty is a very real challenge. We need to have very good data on the P in the inputs and outputs. And pre-1950, we know it was manure, but we don't know, for example, how much the P content was or the percent moisture. Same for the rock phosphate. So as a result, we've got these confidence intervals. This is the gray color around the solid colored line. We've got large uncertainty around these balances. And typically, lower balances in magnitude tend to have lower uncertainty. And the easiest systems, are those with no inputs in this example, because then the only output is grain, corn grain, and we have a pretty good sense constrained of P content of corn grain. The other point that I want to illustrate is that of soil test P values, again, in ag context. Soil test P simply refers to agronomic tests. These are extractions. They're offered by commercial labs today, so they're accessible, they're rapid, large source of siloed data. Think of all the millions of observations of melic P across the US Corn Belt, but we can't access it because it's privately held. I want to emphasize this is not a measure of available phosphorus. Soil tests were done to help predict the probability of a crop yield response to adding more P fertilizer. And people today misinterpret these soil tests as available. They're not. And that's why they're really poor predictors of P uptake. So with that out of the way, shown here on the right is a snippet from the paper where we compared in soils from the Morrow plot archive, soil samples as far back as 1913 through present day almost. We looked at the relationships with melic P today with the stocks that we measured today. And this is the delta stock on the bottom compared to the unfertilized control at the beginning of the study. So this to me right here would be the true residual phosphorus for the delta of the stock. And the point to make here is that we see very poor correlations in terms of predicting the actual amount of residual P stock. So it seems like the soil test PR may be okay for a qualitative evaluation over time. If your soil test value goes up or down, you can anticipate that the residual P is going up or down. But to quantify it with confidence, that's a different question. The final thing that I'll end on from this paper is we looked at data from the Marl plots and from Rothamsted, as well as a few other sites like the Arcadia study in Quebec. And we replotted some of the data that has been collected on the forms of phosphorus. These are operationally defined fractions by the Headley sequential fractionation. The method has all kinds of issues and still provides us at least some standardized assessment, and especially the pools of calcium P and organic P are probably decent confidence. The other ones are perhaps questionable. But that said, it offers a relative examination of changes. So I want to walk you through this graph because I think it shows a very important point that we, I think, report for the first time in this piece. And that would be that if we think about the change in soil phosphorus, so the change of the residual phosphorus, or rather where it's located, the percent of the positive P balance under 3 crop rotations of the Marl plots, maize, soybean, maize, oat, hay, the percent of the residual P is largely found in the surface depth, specifically in the surface 30 centimeters. And note that we have a negative percent, meaning that we saw a net increase in total phosphorus stocks at depth. That we saw a net decrease in the P stocks at depth and a net increase in the surface. That's where we have a negative percent of the residual, of the positive balance. And note too that the different fractions of phosphorus also differ. So there's a few insights from the Marl plots here. One is that in these 3 examples where we have a large positive P balance, a lot of residual P, that the form it takes is not the same. The proportion of the residual P, for example, as calcium P in orange, differs by not just the treatment, but the amount of residual phosphorus present. Second is that we tend to see most of this extra phosphorus accumulate in the surface depth, even though concurrently or concomitantly, we have depletion of phosphorus in the bottom depths. So this is a case for doing full depth sampling beyond surface depths to get a more accurate assessment of total P stock change, which can be used in long-term studies or the chronosequence approach to get a more accurate estimate of the how much. The second example here is from Rothamsted. We have the same depth axis just to emphasize that there was just one depth here. Good example of how you've got a negative P balance for this treatment because it's unfertilized but cropped. And if you have a positive P balance of similar magnitude between farmyard manure versus P plus K, the speciation, or let's say the fractions, look very different. So there is a bit more total residual P in this treatment versus this one, but completely different forms of phosphorus. Even while we're depleting other fractions of phosphorus, we have increases in different fractions. So this is important because it sheds light to what is the form of the residual phosphorus. Keep in mind, residual phosphorus is simply a magnitude. It's a how much. What kind of phosphorus that is apparently a little bit different. And likely soil type, management practices, the form of P input all together conspire to shape what the P is in the soil. It also matters how we measure this over time. So shown here on the right is a study from Rothamsted. We've got the same treatment. So this is the same exact data now shown here. And we split the data into 3 different time points. This data was published over 2 windows from 1903 to 74 and 74 to 93. And then we even have for one treatment going back further than that. And the point here is that depending on how you group these treatments, that when you look at these treatments over time, we see that there's different changes in the P fractions as the phosphorus accumulates. So for example, we see that there's initially a net positive balance, meaning that we have residual phosphorus in the first phase of this treatment. And then in the next 2 phases, we have a large decrease, a net negative balance, hence the negative percent. The point here is that the time scale matters. And that goes back to the opening argument that any mass balance is going to be a time function. So to summarize, Residual phosphorus is a defining feature of the 21st century. It's why we propose in the title as part of the legacy of the Anthropocene in which we find ourselves. Again, this residual phosphorus results from the accumulation of phosphorus where inputs exceeded the outputs at a given system and time scale. Common approaches that can be used and preferably combined to account for the flaws of any individual approach are mass balances, where we define residual P as a positive mass balance, as well as long-term experiments like what you just heard, the Morrow plots, Rothamsted, etc. Soil test P trends, which there's a lot of data out there on soil test P, but it's largely private. And what we argue is the least used approach, space for time, or the chronosequence approach. Next steps that we propose. First is data gaps. There's a lot of data out there, a lot of work to be done to unify and integrate existing data sets on pool sizes and flux sizes. Second is that we should measure better. We need to better standardize how we as scientists report data that will help construct mass balances or other approaches to getting at the magnitude in question. And third is quantifying uncertainty. We need to prioritize how we monitor and manage these resources of phosphorus. Residual phosphorus is a resource. It can be mined by crops and used in the future. Also, we're talking about resources in the other sense of to measure where and how much takes a lot of, well, labor, time, and funds. And I think as important is we need to put the uncertainty at the forefront of discussions that help inform policy. We may not know exactly how much or where the residual phosphorus sits in a field, in a watershed, in a country, and that's important so that policy can account for the uncertainty. We just don't really know at the moment. It needs to be more prominent, I think. Greater needs. Ultimately, scientifically, I think a good next step is to go beyond the magnitude. Remember, residual phosphorus is a net positive balance. It's a how much. That says nothing. It is agnostic to the form of that residual phosphorus. And as we just saw from those two examples, the fractions and thus these pools that we might infer from the fractions of the residual P, they can differ quite a bit. And they're decoupled from the inputs of phosphorus that led to that net accumulation over time. So figuring out the form of phosphorus is important, as well as where it is at finer spatial scales, so that we can better utilize it, for example, drawdown by crops, but also help predict the potential leakage of this old phosphorus, as some call it, to water bodies.
Well, thanks, Andrew, for walking us through that. I know you have another thing you have to get to right now, so I'll hold off on the questions this time. I'll be very interested in seeing what your group comes up with in terms of recommendations for best measurement practices going forward. But we'll hold that discussion for next time. So thanks again for joining us for Phosphorus Science Now.
Sure thing, Matt. Thanks for having me back.
Thanks.